Start my recording. It's been brought to my attention. I didn't post the link for last week's or last. Is it just Thursday or is it both? Let me check. Oh, it's Tuesday and Thursday. Okay. Well, I think that one of them I can immediately adjust and throw that link on there. The other one, I'll upload the video in a little while. I don't want to do it right now while I'm streaming. So 4375. Okay. Um, I'll nope, I don't have that one on my YouTube channel. I will get those uploaded. I'll get all three of them uploaded within, you know, 20 minutes of our class finishing today. Okay. Sorry about that. So I apologize there. So what we are working, I had a question about the homework. It says to write it on a piece of paper. Does that mean it can't be on my tablet to write down my answers? Also, is it okay to write down all of the homework on the same paper, then just submit it to each homework section? Okay, so great question. Um, I want it written and you can write it on a tablet. I don't want copying, pasting, and I don't want typed responses. So, um, if you want to write it on your tablet, that's just fine. I just want you to know your own, like your own penmanship using the stylus or whatever. Follow-up question to that was, is it okay if I write down all the homework on the same paper, then submit it to the each homework section? I'd like you to be able to kind of segment each piece off, just because if it's, eight assignments and you submit homework.pdf to all of them, that's going to, then I'm going to have to like scroll through every time that I go to each homework, or sorry, whenever I open your homework, then I'll have to scroll to like page 7. Does that answer your question? Does that make sense, first of all? And does that answer your question? So, okay, cool. Um, I am recording. So what I need to do is I need to post last week's videos. I will do that. That will not take much time. Um, Today, what we are going to be talking about is we are going to be talking about protein analysis, and we're going to get into DNA. The slide deck that we are using or the slide deck that we are looking at right now is week 10, day 1. The DNA one that we are going to get to at the end of this class period is week 10, day 2. Okay. So I'm going to scroll back just to kind of give you an overview of what we talked about. Actually, before we do that, last class period, we talked about columns. And we talked about different types of columns. There's a word I'm looking for. It starts with a CH, that it's column CH. Finish that word. Could somebody add to the chat that kind of idea? Chromatography. That's exactly what I'm looking for. So last week, one of the things that we talked about were different types of column chromatography. And chromatography is used to separate different things. So when you do chromatography, you basically have a stationary phase and a mobile phase. Our protein is in the mobile phase. The stationary phase is what kind of selects four different molecules. Um, what we were getting at was, how do you take a complex mixture like a cell, like a tissue extract, and pluck out individual proteins? We talked about different types of column chromatography. For example, we talked about cation exchange. I'll just say cation X. Anion X. Gel filtration or size exclusion or size X. And affinity chromatography, which I'll just characterize or describe as FX. Okay, now I'm going to go with A, B, C, D. In the chat, tell me the letter that corresponds to the chromatography column that has small beads with pores in those beads. So here's a kind of visual of that bead, and here's one of the pores that a protein can fit into there. It is size exclusion chromatography. Awesome. Okay. Now, here's a bead, and I'm going to chemically modify that. I'm going to permanently modify this. And what I'm going to do is I'm going to add a amine to it. And that amine's going to have, there's two, then H2. It's going to be positively charged. Could somebody tell me what type of chromatography, A, B, C, or D, will use a bead like this? Okay, so the bead itself, like this thing that I've written the NH2 with a positive charge, that is permanently there. So think about what is that going to attract? I'm getting a peppering of answers. I'm getting lots of A's, lots of B's, and now I'm getting lots of B's. And you are absolutely correct. This type of bead is valuable for anion exchange chromatography. The reason that it's anion exchange chromatography is because it is going to attract negatively charged molecules. So it's going to attract anions. Okay, now the last one. So then if I were to do this, here's my bead and here's my group. Um, that has a negative charge. This is going to be good for cation exchange chromatography. Perfect. Now, the one that I'm not, well, I mean, process of elimination, if you were to have a bead that looks like this, and then we had a glucose molecule or a sugar on it, this is going to be valuable for attracting proteins that have an affinity for glucose. So that would be your affinity chromatography. Could you give me a 1 if you feel good about the different types of chromatography, or a 3 if I really can't keep them straight? Awesome. Awesome. Okay, great. Thank you very much. So the type of chromatography is based on what they attract. That's a great question, great observation. When it comes to anion and cation exchange, yes. Okay. So, what we talked about last week was we focused on separation techniques. The important thing about affinity chromatography and all of our different types of column chromatography was all of the proteins that are right here, we can reuse those. We can collect them from this tube and go ahead and use them again. Now, what makes this different from this right here, gel electrophoresis, is when you do gel electrophoresis, you use this molecule, sodium dodecyl sulfate. And that basically coats the protein with negative charges. So your protein has a uniform negative charge on it. And that allows you to purify a protein and separate them. And so then you can say, oh, well, here's my complex mixture. I'm going to put a star by the one that I'm referencing. Here's my, oh, here's my complex mixture. There's tons and tons of different proteins in there. I go through a couple of different chromatography steps, and lo and behold, I get my thick band right here. Okay, awesome. There's my protein. The thing is, this right here, that protein, we can't really use that anymore because that protein has a bunch of this molecule plastered all around it. Okay, so when you think about separating proteins based on size and you want to use them again, you want to be doing gel filtration or size exclusion chromatography. Gel electrophoresis, shown right here, does separate based on size, but you don't get to use those proteins again. Could everyone give me a 7 if, okay, I follow that, or a 3 if I've still got a question about that? And if you've got a question, please don't hesitate to follow up with what that question is. If you're like, I need something to like hang my hat on as a detail, I would think chromatography, I always kind of think about it as a preparative step. You do analyze and get some information from it, but you're preparing your protein, you're purifying it from all of the other junk. Whereas electrophoresis, that's more just an analytical step. Okay, so you can basically just analyze it. You're not really going to be able to prepare it to do something else with it. Now, the point here on this slide was really just to show, hey, the migration distance that a band has, or sorry, How far a band migrates relates to how large that protein is. So that's a value of SDS page. Isoelectric focusing, also known as IEF, is a technique that you can use to separate proteins based on their pI or their isoelectric point, their overall or what pH do they have to get to an overall charge of zero. Now, I mentioned this 2D electrophoresis because this is a technique that's really powerful and it's something that's old, but it's kind of a fundamental thing, and you're separating based on size and by PI. And when people use this to analyze and compare a healthy or wild type cell to like a cancerous cell line, you can make some pretty significant discoveries. Oh, well, why is this protein present in the wild type but absent in the cancerous type? Is it getting degraded? Is it getting modified? What's going on there? So those are sort of questions that you can answer. So last time what we talked about, chromatography. We talked about isoelectric focusing or IEF, SDS page, and even before then, we talked about we talked about specific activity. If you are purifying a protein, do you want your specific activity to be H high or L low? Add it to the chat. Absolutely. The higher your specific activity is, the more pure your protein is because you're specifically looking at one protein. Now, when we were talking about specific activity, we talked about the Lambert-Beer law, and I'm going to bring that back because it's relevant to proteins. And I gave you this hypothetical, oh, well, we could observe wavelength of this, and that tells us our enzyme is present. Well, you can use the Lambert-Beer law, which is shown down here, A equals epsilon CL, A equals ECL, to tell you a little bit about the amount of protein in a sample. And the reason that you can do that is because proteins typically have UV absorbance maxima around 275 to 280 nanometers. Now, if you're like me and you think, wait, that doesn't make sense, those numbers, wait, I always have a tendency to think about 450 to 700 nanometers as being an important range. Could somebody tell me what this wavelength range is? Exactly. It's the visible light range. For some reason, those numbers stick in my head, but anything outside of that doesn't. So is 275 to 280 in the UV range? Yes, it is. So you can use UV light to detect protein. How do you do that, though? Well, proteins have unique shapes. Does it have to do with the shape of a protein? No, what it has to do with is three amino acids. Tryptophan, single letter code W, three-letter code of TRP. This is our molecule that basically has a five-membered ring. Wow. And a six-membered ring right next to each other. So we've got a double ring structure there. That's not a very well-drawn R group. Tyrosine. Tyrosine has a single letter code of Y and a three-letter code of TYR. TYR, Joel. And that R group looks a little bit like this. Chew, 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 and then OH. I always think about it as a tire, like a bike tire. Tire, oh yeah. Okay, so there's tyrosine. And then last but not least is phenylalanine with the single letter code of F, three-letter code of PHE, and an R group that looks a little bit like this. Now, with that in mind, what do you notice about all these R groups? Well, they all have rings. They're all our, Melanie, you stole the word right out of my typing. Aromatic. These are our aromatic amino acids. So essentially what that means is that if your protein has an aromatic amino acid, you have a mechanism to detect that using UV light. Now, would you be able to detect, or do you think that you'd be able to detect a protein that doesn't have tryptophan, tyrosine, or phenylalanine at all? If you looked at how much light does it absorb at 275 nanometers or 280 nanometers? You think you'd be able to detect that? Chances are pretty slim. But do you think every protein or most proteins have a little bit of each of these? Yeah. So statistically, you're going to be able to detect a protein using the aromatic amino acids and the presence of those aromatic amino acids. Now, if you have a protein with tons of these aromatic residues, it might absorb more light. Now, or absorb light at 280 nanometers. So, tryptophan, tyrosine, phenylalanine, those are the amino acids that are going to absorb a little bit of light in the UV range that give you a way to determine if there's protein present in a sample. Could everyone give me a T if that sounds good, or a M if meh, doesn't make sense? Okay. So, essentially, what that means is our aromatic residues give us a way to determine total protein. Hmm, total protein. I guess we're going to go on a little bit of a journey through time. Have we seen total protein or have we talked about quantifying protein before? We have. And this dates all the way back to when we were looking at this table right here. Total protein. So when you see this column, I want you to think about it as, oh, this is determined from aromatic residues. Now, what about units of enzyme activity? Well, that's something that's going to be specific for whatever enzyme you're trying to purify. So we do need to use A280 or absorbance at 280 nanometers to detect protein. Now, specific activity is going to be something about your enzyme of interest or the enzyme that you are trying to purify. Now, could you give me a 1 to 5? One being, I feel good about this, five being, I'm struggling with it when it comes to quantifying proteins and specifically quantifying your enzyme of interest. Both of those things. How do you feel about that? Okay. So think about this as I have not given you I have not given you a definition of this is the equation or this is the wavelength that you have to use to determine the concentration of your enzyme. The reason for that is because every enzyme is going to have something a little bit different. Some enzymes will maybe generate a product that absorbs light at, 310 nanometers. Other enzymes will generate a product that is produced and is measured within the visible light range. When it comes to specific enzymes, that's a case by case basis. When it comes to total protein, that's that A280 or 280 nanometers. Okay, now the next thing that we're going to tackle is protein sequencing. So protein sequencing is a process that takes a lot of different steps. Now, there's only a portion of this information that I really want you guys to understand well. And the portion of it to me is kind of like it's like a puzzle. And I really like this part of it. And I hope that you guys will enjoy it as much as I do, or, close to, or maybe even enjoy it more. So, protein sequencing is something that is technically more challenging than DNA sequencing. It's not as abundant or as common as DNA sequencing. And the reason for that is because most of the time, if somebody's analyzing a protein, well, they're going to get the protein sequence from its genetic code. So they'll use the DNA sequence to predict what the protein's code is. Now, there are two ways in which protein sequencing is done. One way is known as Edman degradation. The other is by using mass spectrometry. So between these two methods, this is pretty darn fast. This is a little bit more technical and requires a little bit more time. Now we're going to go over the steps, but there's only a handful of this that I want you to really focus in on, and I'm going to highlight that. So our steps for sequencing. The first thing that you have to do whenever you purify a protein or want to sequence it is you have to separate the individual subunits. So what that means is that let's imagine that we have a trimer. Okay, here's our trimer. And let's go ahead and say that we have an alpha subunit, a beta subunit, and a gamma subunit. Okay, now just to bring back a little bit of information, does this protein have a primary structure, Y or N in the chat? This hypothetical protein? Yeah, absolutely. Does it have a secondary structure? Does it have secondary structural elements? Yeah, probably. Does it have a tertiary structure? Yeah, pretty reasonable. Does it have a quaternary structure? That's really the question that's kind of the like decider. This protein right here, by being made up of three subunits, it does have a quaternary structure. If I compared it to, let's say, this singular protein that is only one subunit, this one right here, no, I'm going to do a 4 and then that not symbol, no quaternary structure. I know that this is some older stuff, but I just wanted to kind of bring it back. So the first step, whenever you're doing any sort of protein purification and analysis of the sequence, is you've got to take this trimer and you've got to break it into individual subunits so that then you can isolate them and separate them from one another so that you can get an alpha sequence, the beta subunit sequence, and the gamma subunit sequence. Because if you don't disrupt the interactions here, you're going to get one long sequence that it's going to be really difficult to like piece it together. The way that I kind of think about this is if you are, it's almost like if you had, and this is kind of a weird thing to imagine, but let's imagine that you had three puzzles. Yes, three puzzles that you put together, and they're overlapping like this. And you wanted to disassemble and reassemble all of these individual puzzles. If you took these apart and put them into all of the pieces into one bag, is it going to be difficult to reassemble this puzzle as is? Yes. If you were to take this puzzle, take it apart, or remove it from that, then take it apart and put all the pieces into this bag and then try to reassemble it, is that going to be easier? Yeah. Okay. Now, on a scale of 5 to 10, 5 being, oh, perfect, nail on the top of the head, and 10 being, what on earth are you talking about? How is that puzzles analogy? Kind of anticipating a bunch of 10s. I hope I don't see them. But, uh-oh, now I'm not getting any responses. 7. Okay. Thank you. Thank you for your honesty. Okay, so back to the point though. Whenever you're doing any sort of protein sequencing, you want to separate each of the subunits, however many there are, into individual kind of extracts. So you separate the subunits. That's the first step. The next step is to break any disulfide bonds. Could somebody tell me the single letter code for the amino acid that's capable of making a disulfide bond with another of itself? Add it to the chat. What is our single letter code there? C. Absolutely. Cysteine. You got cysteine residues. You got to break those disulfide bonds. And those are unique. We talked about breaking those. You use beta mercaptoethanol or BME or DTT, dithiothiotol, because those covalent bonds have to be reduced. After that, you're going to get the amino acid composition. You're going to find out what are the amino acids that are in here. Then you're going to do something known as N group analysis. And then this step right here, number 5, molecular scissors. That's what we're going to focus in on. You're going to use molecular scissors to break up these individual peptides. Step 6, you're going to repeat that as much as possible with different molecular scissors so that you can reassemble your sequence. And then you'll sequence each purified peptide fragment using Edman degradation and reassemble it. Sorry, step 8 is reassemble it and reconstruct it. Now, a visual of that. Here we've got a pair of peptides. Would this protein right here, as I point to it, would this protein right here have a quaternary structure? Y or N to the chat? Y for yes, it would have a quaternary structure, N for no, it would not. Absolutely. It will have a quaternary structure because this blue peptide is one peptide and the green one is a separate one. They're connected through a pair of disulfide bonds, or a pair of cysteines that make a disulfide bond. So you want to separate these two. Then what you're going to do is when you have each one of these peptides separated, you're going to do some sort of molecular scissors action. You're going to play around with some different chemicals to break those into individual shorter peptides that are a little bit easier to analyze. And whenever you break those down, you can then begin to reconstruct them. Okay, now I'm going to kind of jump ahead. I'm going to scroll through the next little bit a little quickly. Um, First of all, separation of subunits is done by extreme pH, urea, guanidine HCl, high salt concentration. So I want you to think about this as what are the conditions in which you would use to separate subunits? You'd use some extreme conditions. Those being extreme pH, like extremely basic, 14, or 1. You'd use 8 molar urea. Urea and guanidine hydrochloride, these two compounds, they're chaotropic agents. They're going to disrupt hydrogen bonds. High salt concentrations can also disrupt hydrogen bonding. I want you to know that the extreme conditions are what you're going to use to separate individual subunits. Disulfide bonds, I want you to know that DTT and beta-mercaptoethanol will be used to disrupt those. We've talked about those previously, so that's kind of why I want you to know. Now, I'm not worried about you knowing iodoacetate or anything like that. So what I want you to know is that there are extreme conditions that are used to disrupt or separate individual subunits. Disulfide bonds are broken with DTT and beta-mercaptoethanol. And then what I want you to know is basically jump on to step number 5. Step 3 is where you determine all of the amino acids that there are present in that protein or in those peptides. Step 4 is a way that you could determine C-terminal and N-terminal amino acids. But now, step 5. Okay, you might be curious, why are we jumping, or why is step 5 so important? One of the reasons is we've talked about trypsin and chymotrypsin. Could anyone tell me what trypsin and chymotrypsin are? And I'll give you a little bit of a hint. We talked about these in the context of the catalytic triad. And if you can't remember the class of enzymes these are, you could tell me maybe what type of bond do they break? Add it to the chat. Sorry. So trypsin and chymotrypsin, these are two examples of what are known as proteases. These are enzymes that break peptide bonds. Could anyone give me a 1 in the chat to show that you're here? Okay, cool. Okay, so trypsin and chymotrypsin are proteases. These are enzymes that I know that I used this term last, or when we talked about these previously, I mentioned them as being kind of cannibals because they are going to clip up and they're going to digest enzymes themselves. Now, if we look at this right here, I want you to follow along with me and say that this is the N-termini of an amino acid. This is our alpha carbon. This is our C-terminal of an amino acid. This is the N-termini of the next amino acid. C, C, N, C, C, and I'm just going to put an R here, meaning it just kind of goes on into eternity. Okay, now this right here is our peptide bond. Could you give me a 5 in the chat if you're good with that? You understand that? Okay. Now, when we talk about proteases, also known as endopeptidases, there are 3 that I want you guys to know: trypsin, chymotrypsin, and elastase. These are all proteases that cut peptide bonds at specific locations. Okay. So what that means is they will look at, say, the primary sequence of a peptide. And I'm just going to, whenever I show the primary sequence of a peptide, I'm going to just list I'm going to put a star to indicate my N termini, and then I'm going to put the single letter codes. Is there anything specific you want us to know from the other steps and slides? Yeah, I want you to know the reagents that are responsible for breaking disulfide bonds. I want you to know that extreme conditions are necessary for disrupting interactions between individual peptides, and that's kind of it. So I think that, yeah, that's my answer there. Good? Okay, cool. All right, so what a protease is going to do is it's going to see a peptide, and it's going to look at the N termini, and then it's going to say, okay, well, the first amino acid here is an alanine. Then a W for a tryptophan, K for a lysine, L, a R, and a P for a proline. So this is my N termini, and then I'm going to say N term, and then this down here is my C term. Okay, I'm going to erase this star. I'm going to erase that arrow. So here is my peptide, though. Now, what a protease is going to do is it's going to look at this, and it's going to see, okay, there's a peptide bond between A and W. There's a peptide bond between W and K, K and L, L and R, R and P. Now, if this is the C termini, is there a peptide bond? If this is where it ends, is there a peptide bond there, Y or N? No, there's not. Okay, great. Now, let's imagine that chymotrypsin was our enzyme. Chymotrypsin has a rule, and it has a specificity. What chymotrypsin is going to do is it is going to look at this Rn-1 position. Okay, this is that Rn-1. Could you tell me, is that to the left or sorry, is that on the N termini or the C termini end of my peptide bond in this model right here? So this is the N termini, and then this is the C termini. Can you give me a 1 in the chat if you follow that? And any other character if you do not follow that. Okay, so chymotrypsin, it's going to jump onto let's just imagine that I'm going to use Pac-Man. There we go. There is chymotrypsin. It jumps onto this and it says, okay, here's the N termini. What I'm going to do is I'm going to process this molecule going from the N termini to the C termini. Chomp, chomp, chomp. Okay. I am looking for a amino acid residue on the N termini of a peptide bond. I am looking for either a phenylalanine, a tryptophan, or a tyrosine. So essentially what that means is that in this location, right here, if there is a phenylalanine, tryptophan, or tyrosine present there, chymotrypsin is going to say, okay, cool. I'm going to cut the peptide bond after that residue. Could you give me a 9 in the chat if that makes sense? And any other character if it doesn't. Okay. Now let's go ahead and translate these. Phenylalanine is F, tryptophan is W, tyrosine is Y. Let's imagine chymotrypsin jumps on here. Chymotrypsin's going to look at this peptide. The N-terminal amino acid is alanine. It's going to say, okay, alanine, I don't care about alanine. Could you give me a 6 in the chat if you understand that chymotrypsin doesn't care about alanine? Okay. It's going to say, I don't care about alanine, but I'm just going to go ahead and cross through it because I'm done with it. The next amino acid is tryptophan. Does chymotrypsin care about tryptophan? Give me a T in the chat if it does. It does. Is chymotrypsin going to cut, when it sees the tryptophan, is it going to cut the peptide bond that follows it or the peptide bond that precedes it? So my question is essentially, is chymotrypsin going to cut here or is it going to cut here? Give me an A for this one and a B for this one. Is it going to cut A or B? I'm sorry for drawing over stuff. It's going to cut B. You're absolutely right. Could you give me a 9 in the chat if you follow that it's going to cut B? And a 3 in the chat if, no, I'm mixed up. I thought it was A. Okay. Now, what chymotrypsin is going to do, I'm going to erase some of this because this is getting kind of messy, and I apologize for that. What chymotrypsin is going to do is it's going to take this peptide and it's going to cut at B. So I'll erase A, it's going to cut at B. And what it's going to do after that is it's going to release this AW. That is basically what is found prior to that peptide bond. So it cuts the peptide bond, and then what does it have left? KLRP. Now, my question for you guys is, does chymotrypsin care about anything else in that peptide? Is there a phenylalanine, a tryptophan, or a tyrosine there? Melanie says no. Anyone want to second that? Let's see. I second it. So if chymotrypsin was given this peptide, I'm going to start over, AWKLRP. Chymotrypsin is going to cut it into these two peptides, and it's done. Could you give me a 1 in the chat if that makes sense? I understand it based on these rules, or a 6 if that doesn't make any sense. Okay. So now what I want to do is, when it comes to these proteases and these endopeptidases, what they are also called, I want you to know the rules. I want you to know what chymotrypsin is looking for. Chymotrypsin is looking for a F, W, or a Y. What is trypsin looking for? Could somebody give me the single letter codes for trypsin? What amino acid it's seeking? Trypsin is the one at the top. It's looking for positively charged residues like ARG. Single letter code for that is R. And LYS, the single letter code for that is K. Now, I want you to know the specificity because I want you to know the rules, and then we'll get to the exceptions to the rules. So trypsin is looking for R or K. Now, let's go back to this same peptide, the AWKLRP. Y or N, is trypsin going to cut this? Got a bunch of votes for Y. Yeah, I agree it will. Okay? Because what trypsin's going to do, and I'm going to make trypsin a Pac-Man as well, but it's going to be a little bit different. There's its I. Trypsin's going to see this A, doesn't care. See the W, I don't care. See the lysine, and it's going to say, oh, okay, lysine's a positively charged residue. I'm going to cut right here. Great. Let me erase this. So it will release the tripeptide AWK. Could you give me a 7 in the chat if you're good with that? Okay. Now, what that's going to result in is LRP. So this brings up, well, what is trypsin going to do when it gets to this? It's going to see the L. It's going to say not interested. Then it's going to see the R. It's going to say R for arginine. That's a positively charged residue. Okay, cool. I'm ready to cut. And then what's it going to see after that? It's going to see a proline. And what do we know about proline? Could anyone describe the amino acid proline? Use any words in the chat to describe proline. And I'm thinking like scientific words, not stupid. Uh, Lame, boring, strange. What do we know about proline? Proline is our amino acid that looks like this. CC. Remember, it's the ring that kind of swoops back in on itself. So and I remember I always got tripped up with proline because I was like, oh yeah, the N termini, that's going to be positively charged. So the R group is polar. Close, but it's not. The R group for proline is basically these three hydrocarbons. No worries. So proline is a non-polar amino acid. But in addition to that, one of the things that's significant about proline is the fact that the alpha carbon kind of swings back to the N termini. So the alpha carbon and the N termini are connected in two different ways. That makes this amino acid R group very, very inflexible. And it causes quite a few problems. If you remember, this is actually a secondary structure breaker. So proline disrupts secondary structure. In addition to that, I want to draw your attention to this little exception. And I'm highlighting this exception to all trypsin, chymotrypsin, and elastase. What this is saying is that trypsin, for example, is going to cut after an arginine or a lysine unless there's a proline there. So trypsin will see this LRP and it's like, I am ready to cut. I am looking forward to disrupting this peptide because I see an arginine and I love cutting after arginine. But can trypsin cut right here? Wire N to the chat. No, it can't because this is one of those exceptions. So when it comes to the outcomes of this particular peptide, AWKLRP, chymotrypsin and trypsin will both cut it. And they will both release two different peptides. What it all boils down to, the important thing that you guys need to know is what are the amino acids that these different peptides are looking for? I always think about it just with the single letter codes. Trypsin, R and K. Chymotrypsin, FWY. Elastase. Elastase is going to look after or look for alanine, glycine, serine, or valine in that specific position. It's going to cut after those. So now just to apply elastase, AWK-LRP. And if you're looking at this peptide and thinking like, oh, what's the magic of this peptide? There's no magic. I randomly put letters down. My question for you guys, and answer Y or N, will elastase cut this peptide right here? It will. Can you tell me what will result from elastase cutting this peptide? And separate your products with a comma or a semicolon or something like that. Elastase is looking for an alanine, glycine, serine, or valine. Whenever I look at this, I see one of those amino acids. Favor? Absolutely. It's going to cut right here. And that's all. Now, these three proteases, I want you to know their cut pattern because you're going to have questions that ask, well, this peptide was used. What are the products? Now, I've got a poll question for you guys. And this question is, which of the following peptides will be cleaved? Let's see, where are we? Sorry. Which will be cleaved by trypsin? We've got four options. Is trypsin going to cut ARK, AAR, DAL, or LAD? And go ahead and stop this in just one moment. And it looks like we are, yeah, there we go. The results are that, well, 73% of the class predicted that it would be A. We jump back. Trypsin is going to cut after a positively charged residue. In this particular peptide, peptide A, there's a arginine and a lysine. Now, my question for you guys is, when you look at this peptide, or this peptide and peptide B, these both have positively charged residues. Here's ARK, and there's R. Could anyone explain succinctly why they didn't select B? I mean, there are four people who selected it. I'm sorry. Peptide bonds only. Nice. Is there a peptide bond here? There is not. So it can't cut after something when there's nothing to cut. Okay, so we're at 73%. Great job. Now, the next question is this. A peptide is cleaved and results in the following products. What protease is used? Okay, so we'll just kind of work on this one together. We've got essentially three products. We've got Ka, Rg, and K. So we could kind of assemble this in three different ways. It could be Ka, Rg, K, one option. It could be Rg, Ka, K. It could be KRGKA. At the moment, the most popular answer is A. And I want to tell you the way that you approach this sort of problem, because we don't know the order in which these are reassembled. All that we know is that this side is the N termini, and this is the C termini. So this last peptide, I would classify this as a positively charged residue. So is there any way that I could reassemble this and know, or think about this particular residue and know where it goes? Before I do that, though, what do I notice in peptide one, two. I'm going to say one, two, and three. What is on the C termini of peptides one and two? What type of amino acid is that? How would you classify that residue? This is an alanine, and this is a glycine. Could you give me a one in the chat if you agree with that and you follow that? Josh said nonpolar. Josh is spot on. Alanine and glycine are both small amino acids. Okay. Um, do we have a protease that cuts after small amino acids like alanine and glycine? We do. Elastase. So elastase will cut after alanine and glycine. Okay. So essentially, and follow me on this, please. What information does that tell me? That tells me that the order of this is either of this reassembled peptide is one, then two, then three, using these, or it is two, one, three. How do I know that this one is at the end? The way that I know is that if this peptide, if this K was anywhere but at the end, then I would be cutting on the C termini of a positive residue. Does that make sense? Give me a 5 in the chat if that makes sense. Okay. Okay, I've got lots of 5s there. Melanie said KKA instead of KRG. Okay, so if it was, well, I know that KKA is impossible. I know that that is not an option because I would have to have trypsin cut on the C termini of this lysine. And if I look at my other two peptides, do I have a positively charged end or a positively charged residue on the C termini of peptides one or two? K can't be at the beginning, is what I'm saying. Okay, so let me give you a couple of options. So we could take these products and I'll reassemble them. I've got KKARG. And if there's another permutation that I overlooked, please tell me. Another option is KRGKA. Okay, so those are both where I have peptide number 3 at the beginning. That is 3, 1, 2. So that's 3, 1, 2. Okay, then we've got, let's see, KARGK, let's see, KAKRG, and then let's say RG, KA, K, and RGK, KA. Okay. Now, does everyone follow? These are some of the possible permutations. I think that might be all of them. Could you give me a 7 in the chat if you see? Okay, yeah, I can see how those are the organizations. Okay. Now, what I get if I were to use, let's say, trypsin to digest this, trypsin's going to cut after a positively charged residue. So if trypsin was my peptide, how would it treat this first one? Well, it would cut here, it would cut here. So my product, and then it would also cut here. So my products, if trypsin were the protease, would be K, K, A, R, and G. So is this a possible option? No. The only way in which we could reassemble this is we couldn't use trypsin. In addition to that, we have to have this K at the very end because it's a positively charged residue. So what that all boils down to is this is an option and this is an option. And elastase is the one doing the cutting right here and right here, or right here and right here. So if this question were instead to ask you, well, what are the possible outcomes? Or what are the possible original sequences? The only options that are viable are KARGK and RGKAK. Does that make sense? Give me a 5 in the chat if that makes some sense and you follow that, even if this kind of got a little all over the place. Okay. Now, what I want you to do is, or what we're going to do here is kind of look at a possible product or an example of this. So this is a really good practice exercise. What this shows us is we have a single polypeptide. And this would be a problem where you get all of these individual peptides from a certain cutting, and then you reassemble it. Okay. So the story here is that you have a polypeptide and you get all of the information about all the amino acids that are present. Okay. At some point, there's a analysis of the ends of the amino acids or ends of the peptide, and you find out that glutamic acid, is the N-terminal residue. Okay, so this is a piece of information that you will be provided. E is at the start. Okay. Then, after cleaving this peptide with trypsin, you get these 4 peptides: T1, T2, T3, T4. Your object and your goal with this is to reassemble this peptide into the correct sequence using the information that you have. Now, the first piece of information is that you know E is the N-terminal amino acid. Could you tell me T1, T2, or T3, which of these is your N-terminal peptide? Add it to the chat. T2, you're absolutely right. And I'm going to scribble out this answer at the bottom. I mean, I don't really care. It's the point is going through the exercise. So we know that T2 is the first peptide. How do we know that? Well, because we were given some information that the N-terminal residue was a glutamic acid. Lo and behold, T2 is the only one that begins with glutamic acid. Okay, so now we have a couple of options. This protease could, or this product could be T2 followed by T1, T3, or T4, or it could be T2, T3, T1, T4, both good options, or it could be T2, T4, T3, T1, and so on. So at this point, all we know is that T2 is the N-terminal peptide. Give me a 7 in the chat if you're good with that. Okay. Now, we know that after we cleave this with trypsin, these are the products that we get: T1, T2, T3, T4. Could anyone tell me definitively which of these peptides, which of these 4 peptides is at the end, at the C termini? Is it, well, it's not T2. So is it T1, T3, or T4? And then answer why it is that. We know that trypsin is going to cleave after a positively charged residue. When we look at these peptides, T1 ends with a positively charged residue. T3 ends with a aspartic acid. T3, Douglas, you're absolutely right. T3 is at the end. So essentially what we can do is we can say that our peptide is T2 followed by something, followed by something, followed by T3. Because T1 and T4 both end with positively charged residues. So that means that trypsin can cut after them. T3 ends with an aspartic acid. So that means if it ends with aspartic acid, trypsin can't cut after it. Therefore, it has to be the C-terminal amino acid. Could you give me a 9 in the chat if you follow that, or anything else if I'm a little bit mixed up? I follow T2, but I don't understand T3. 8.5, okay. So with this information, whenever you're told something like it's cut with trypsin, you got to know the rule of trypsin. Trypsin is after a positive residue. Now, the next piece of information. So this would be like, if this is where the question ended, all that you could do is you could say that it's T2 followed by T1 or T4, and then it ends with T3. You don't know if it's T1, T4, or you don't know if it's T4, T1. Okay, so I'm going to erase this right here. Now, what is done is you cleave another copy of that sample with a different protease. And in this case, we're going to cleave with cyanogen bromide. I didn't ask you to know cyanogen bromide, but up here, I would tell you something like cyanogen bromide will cleave at methionine. So at the end of methionine, on the C-terminal end of methionine. So what we're going to do is we're going to look back at the other information that we have. C1, C2, or C3, based on our N-terminal information, which peptide is the N-terminal peptide? C1, C2, or C3? We know that we're starting with a glutamic acid. Is that C1, C2, or C3? Yeah, C1. Okay. Now, the next piece of information is cyanogen bromide cleaves after methionine. Does C1 end with methionine? It does. Does C2 end with methionine? Yes or no? Y or N? No, it doesn't. So what does that tell us about C2? Is it in the second position or in the third position? It's got to be in the third position, C2 over here. So what does that mean for what's in the second position? Well, it means that C3 is in the second position. Now, why not last? Great question. The reason that C3 is not last is because, well, it ends with a methionine. So there has to be something after that. Now, if we look at C2, we can compare. Since it has a D as well. When we look at C2, we know that since it doesn't have a methionine at the end, that cyanogen bromide couldn't cut after it. So the fact that it has, so then what we can do is we can also compare the information that we have. We concluded first and foremost that T2 was our first, or was our N-terminal peptide. Give me a 7 in the chat if you're good with that, if you agree with that. Going back to our trypsin information. Okay. And then, you know what? Let's just kind of piecemeal this. We also agree that C1 is our N-terminal amino acid, or our N-terminal peptide, because that starts with E. So you can follow the sequence, E, G, A, Y. Okay, those first 5 residues. EGAAY, HDFEP, HDFEP. Should these be completely aligned? Yes, they should. Next is IDPR, IDPR. Oh, and then G, okay, G. Does anyone see GAS in another peptide, in the trypsin peptides? Tell me which one. T1. So GASM, GASMALIK. Now, can you look at C1, C2, C3 and tell me, is there a peptide that starts with ALIK? C3, ALIK, YLIA, okay? Where is this region overlapping in our trypsin digestions? T4. So I've got T1 or T2, T1, T4, and then YLIA, CGPM, and then CGPM, TK, TK, DCVHSD, DCVHSD. Okay. Could anyone see how, yeah, there's a little bit of a gamification there. You use information, you overlap it. You go with this information, you volley back and forth. At the heart of all of this, what you need to know is, well, what does trypsin cut after? If I gave you a question like this on your exam, do you think you'd be able to answer this? Y or N to the chat. Okay. I would recommend going over this just so you have a little bit more comfort with it. Okay, now we are going to switch gears and we are going to go on to DNA. And so I'm going to end this slideshow, keep those annotations. So when it comes to protein sequencing, the most important stuff is going to be those molecular scissors, understanding what they cut after and all of that. So there we go. Full screen. Now I know that many of y'all have had DNA, whether it's in a biology class or something else, but we're going to go into some of the differences and nuances. And hopefully you'll learn something new, and hopefully there won't be anything where you're like, that doesn't make sense. Okay, so nucleotides and nucleic acids. What's the big deal? DNA, deoxyribonucleic acid, RNA, ribonucleic acid. Hershey-Chase experiment. What did the Hershey-Chase experiment do? Could anyone sum it up real quick in their chat response? Or in a response in the chat? The Hershey-Chase experiment? Oh, I don't remember. Sounds familiar though. I like it. Showed virus-infected cells with their own DNA. Okay. In a more general sense, what the Hershey-Chase experiment did was it proved that DNA is the carrier of genetic information. So the, sorry, DNA is the carrier of genetic information. On the left-hand side, what we have is bacteriophage that infected bacteria. And what was labeled in these cells was, and I'm sharing the, good. We had radio-labeled or sulfur, we had radio-labeled sulfur in the capsid protein, so the protein that made up the structure of the virus. The DNA was not radio-labeled on the left-hand side. Okay, and so what happened was these bacteriophage infected the cell. Great. Then they centrifuge separated the virus from the infected cells, and they found that there was no radio label in the cells. Okay, so that transfer of genetic information did not take place here. Or sorry, no transfer of the radio label took place here. No radio label transfer. Next fur. On the right-hand side, phosphorus, P32. Phosphorus 32. There was a radioactive isotope of phosphorus that was used. And the reason that that radioactive phosphorus was used was because, well, if the radio label stayed with the cells, the only place that that phosphorus is going to be found is in DNA. And so they had these bacteriophage infect the cells with their DNA, with their radio-labeled DNA. And then when they did the centrifugation, separate the virus from the cells, they found that, well, all of the cells had P32, the radio label. Okay, so this was proving DNA carried genetic info. All right. Now, next up, and that's where our next poll question is. In the upper left-hand corner, who is this woman? We got Rosalind Franklin or Eleanor Roosevelt. I'm going to be very surprised if somebody doesn't select Eleanor Roosevelt. And there it happened. There it goes again and again. Six people have selected that is Eleanor Roosevelt. Feel like if one more person does it, you're trolling me. 6. I'm going to let it go for another few seconds. And oh, there we go. Okay, thank you. That was my validation that I was being trolled. Yes, that is not Eleanor Roosevelt. For whoever said, thank you very much. Yes, that is not Eleanor Roosevelt. I am completely confident that 7 people were trolling me. Instead, that's Rosalind Franklin. Now, Rosalind Franklin is responsible for this figure right here, which is an X-ray diffraction image of a DNA double helix. So this work was instrumental in helping Francis Crick, James Watson, and Maurice Wilkins isolate and elucidate, I love that word, the double helix structure of DNA. 1953 is kind of when they published it. Rosalind Franklin's work was done before then. Rosalind Franklin died. I can't remember when it was. I think it was in like 1951, maybe. I don't know exactly. I can't remember. But these 3 fellows, Francis Crick, James Watson, and Maurice Wilkins, were recognized. They won the Nobel Prize. And Rosalind Franklin was not. One reason for that was she had passed away before the prize was awarded. And the Nobel Prize is not awarded posthumously. But also, I think, yeah, there are other factors at play. So James Watson just died a few days ago, at like 100 years old. And I think he's an interesting, yeah, I'll stop there. Okay, so there's a DNA double helix. That was tremendous because up until then, there were other theories. There was a triple helical structure. This structure right here, it seems so like apparent and obvious. Okay, yeah, that's what it looks like. The structures that also existed at the time were much more complicated, or theorized at the time were also much more complicated. But thankfully, because of Rosalind Franklin, who did this work right here, which enabled these 3 guys to stand on the shoulders of a giant and say, hey, this is what we think it is. And you know what? Everything points to yes, that's right. Now I think she's a very important name to be remembered. And it's really unfortunate that she wasn't recognized during her lifetime. At any rate, this structure is something that you're probably a little bit familiar with. Does anyone know who this guy is right here. And you might be thinking, I've played that game Guess Who. Has anyone ever played the game Guess Who? Put a 7 in the chat if you've ever played Guess Who. Yeah, I think this guy's name is Herman in Guess Who. Not really. His name is J. Craig Ventnor. Herman is just the name. If you've never played Guess Who, it's a game where you like look at people's faces, little cartoon characters. Does your person have glasses? Nope. Click, 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 click. And you have to guess who this other person's person is. If that doesn't make any sense, I apologize. I don't have Guess Who with me, otherwise we'd play it. Um, anyway, he looks a lot like Herman from Guess Who in the 80s. J. Craig Ventnor. He was a guy who, in the early 1990s, late 1980s, spearheaded the Human Genome Project. The Human Genome Project was ultimately published in as a Nature paper and a science paper. It was a joint private-public venture where there was private money coming into it and NSF and NIH money, so publicly funded venture to basically, well, advance determining the human genome sequence. This was tremendous because it opened the door for so many different things. In addition to that, even if you might not be like, oh, well, I got my human genome sequenced and it didn't do anything for me, the the revelations that came from the technology that needed to be developed in order to determine the sequence of the human genome changed everything. It's changed medical science in the past 25 plus years. Um, this is something that is continuously evolving because there are parts of the genome that are not completely sequenced. And so it's not like, oh, we're done. Instead, it's like it reached a point where you could publish it and or where it was publishable, and there was enough that was worth saying that, boom, let's put out some papers. Okay, so but what's the big deal with DNA and RNA? Well, first of all, in ribonucleotides, there is a sugar. That sugar is a pentose, and that sugar is um, ribose. In deoxyribose, the pentose sugar is deoxyribose. Lo and behold. What do you think the difference between those two is? I think it might be an oxygen. One has the oxygen, and then the one that's maybe called deoxy is without an oxy. And you're absolutely right. Here, what we have is an example of a ribonucleotide. Here's a deoxyribonucleotide. The difference between them is the hydroxide group right here on carbon 2 prime, or a hydrogen at carbon 2 prime. Now, a nucleotide, these are two terms that are important to know the difference of. A nucleotide is composed of a nitrogenous base, which is this right here, a sugar, which is this right here, and at least one phosphate group, which is this right here. This first phosphate group off of the sugar is commonly known as the alpha phosphate. Typically, if we talk about ATP, there's a beta phosphate and then subsequently a gamma phosphate for adenosine triphosphate. Nucleosides, by comparison, are composed of a sugar and a base only. So a nucleoside has no phosphate. Now, some of these terms we've seen before, and I've talked about them before, specifically when it comes to ATP. Now, ATP, let's look at this. Abbreviations of ribonucleoside 5' phosphates. Okay, a nucleoside. That means a nitrogenous base and a sugar. But then this kind of addendum of 5' phosphates indicates, well, this is talking about a nucleotide. But does it have how many phosphates? If it's got one phosphate, it's a monophosphate. If it's got two, it's a diphosphate. If it's got three, it's a triphosphate. Hence the alpha, beta, and gamma phosphates. These are nucleoside triphosphates, or NTPs. They are adenine, guanine, cytosine, and uracil. ATP, GTP, CTP, and UTP. Deoxyribonucleosides. Again, same basis, adenine, guanine, cytosine, thymine. And if it's a deoxyribonucleoside, it's a DATP, DGTP, DCTP, and DTTP. And that's where I'm going to stop. Slide 9. I will jump back into this on Thursday. I hope you guys have a wonderful Tuesday. Thank you so much for coming. Thank you very much. Have a great Tuesday, everyone. I will see you on Thursday. Deal? If you have any questions, please, I want to help you succeed in this class. Let me know and we can figure out a time to meet. Deal?